Thank you very much, Joel. Yeah, I should return the favor to Danny. I've learned a lot from Danny, and I should say that, yeah, as a designer, still some of my best friends are evolutionary people. Yeah, but our lab is, our lab is primarily interested in the question of how can we design new protein functions. And in order to achieve this, we must develop better tools. Our tools are currently not good enough for that. And just as importantly, we need to understand much more deeply how function is encoded in natural proteins. So in protein design, in protein design, we use we can also print the presentation. Yes, so in protein design, we use a physics-based atomic energy function to generate and rank different sequences and structures. If you have enough computational power, you can obviously use this method in order to design millions and millions of different sequences and structures. But what we and others have found in using such native state energetics is that these native state energetics on their own are not sufficiently accurate in order to design large and complex proteins, such as antibodies and enzymes, which are the primary interest of our lab. Instead, the unique strategy that we've been developing in the lab also relies on sequence information from natural homologs and conformations from the protein data bank in order to guide the atomistic design process. So you can think about our strategy as sort of a hybrid. We call it evolution-guided atomistic design, which combines native state-specific information using Rosetta atomistic design derived from the native state and family-level information derived from homologous sequences and structures. Now, to me today, this combination seems quite natural, but it took us years to develop and actually longer to accept the necessity of this hybrid strategy. Why was it so difficult for us to accept the necessity of evolution-guided atomistic design? The reason is that the expectation, both from us and others in the field, was that all of the information needed for appropriate design is encoded in the native state. But this is true mostly for small and simple proteins. You can imagine, for instance, coiled coils or simple repeat proteins. By contrast, these are some of the proteins we've designed over recent years, and you can appreciate that they're quite large, hundreds of amino acids in size, and have quite complex topologies. It turns out that for these proteins, you must bear in mind that the native state of these proteins must compete with numerous misfolded states. How many misfolded states? We have absolutely no idea. We also, and this is very critical, we have absolutely no structural information about any of these misfolded states. And this is the key point. If we don't have structural information about them, we cannot use atomistic methods in order to rule them out. And this is where this hybrid comes into play. We use Rosetta atomistic design calculations in order to favor the native state, and we use family-level information in order to rule out or limit, to the best of our capability, the misfolded states. And in this talk, I'll show you in more detail, of course, how this strategy works and how we've been able to use it in order to develop quite general and automated methods for design of protein stability and activity. But before that, I'd like also to show you how we came about this strategy. And this all started from our work trying to design antibodies. Antibody design turns out to be a very challenging and still unresolved problem in computational design. The reason this problem is so challenging is that antibodies are very large molecules. The variable domain in itself, which is the area that we are most interested in, comprises 200 amino acids in 2 chains. And you can see that the most interesting parts of the variable domain, these complementarity determining regions, are mostly loop structures. So you can imagine that the sequence and the conformation space of the variable domain of antibodies are astronomically large. And this is why this problem is so complex. The appeal of antibody design, by contrast, is that we have access to thousands of molecular structures of antibodies. Another appeal is that antibodies are largely modular, and this was already understood by Sarastrothia in the 1980s. So because of this modularity, you can combine, take a piece of this gold antibody, let's say from mouse, a piece from this blue antibody from human, and you can recombine them on the computer, join them together, and design the sequence for maximal stability and antigen binding affinity. And in principle, this gives us access to billions of different structures that we can, just by recombining what we see in the PDB. So this excited us greatly. It's conceptually simple, but it turns out to be a very difficult strategy to implement in practice. So again, we were very excited about the strategy. We went, after developing the design strategy, we designed about 20 different antibodies, all of them targeted to human insulin. And we went to the lab to test their binding. But just before testing binding, we also tested their expression levels. And this is what you see here. The expression levels were terrible. This is a horrifying example of failure in computational protein design. I'd never seen anything worse than this before or after. So very, very bad. But over the course of 5 iterations of this design and experimental testing, we got to the point where now all the designs, all the antibodies that we designed, even though they encode more than 50 mutations from any mammalian germline, they're still stable and well expressed. And the nice thing about this approach is that we can actually trace the methods development along the course of this trajectory and realize what were the most important elements that we included in the design strategy that helped us get from this pitiful stage to much better expressibility and stability. And I'll just point out 2 lessons which turned out to be incredibly general for computational design of complex proteins. One of them is related to the relationships between loops and the scaffold. So in the antibody engineering literature, it often appears that the framework of the antibody variable domain is so conserved that you could essentially shuffle CDRs from different antibodies and a framework in order to generate new specificities. And while it is true that the framework is highly conserved, as you can see here, the framework is in green, it's not absolutely so. And you can see by this arrow, this is the same position in 2 different heavy chain domains. And you can see that while this is an alanine here, it's an arginine here, the alanine can pull together H1 and H2, whereas the arginine pushes them apart. And you can imagine that if you were shuffling these CDRs and frameworks, you would get mostly unstable, non-specific, and low stability antibodies. So really long-range polar interactions between the scaffold and the loop are absolutely essential for designing complex proteins such as antibodies and enzymes, as I'll show further. The second lesson came from analyzing sequences of different structures in antibodies. Here, all of these conformations, all 3 of these conformations have the same sequence length. They come from H3. But you can see that if you cluster the sequences according to these conformations, you can see specific and unique conservation patterns for each of them. Whereas the blue conformation typically has an arginine at position 3, the yellow conformation typically has an aromatic. And these family-level data, as I call them here, are really essential to prevent misfolding. You can imagine that if you misencode this, you would get misfolded and inactive, non-stable, non-specific antibody. So these are the sorts of lessons we learned, and using these lessons, we were able to design not perfect antibodies, but atomically accurate, stable antibodies that bound their intended target. So again, antibody design is still something that we're very actively pursuing in the lab, but I'd like to now go back a couple of slides to this slide. When we first saw these data of improvement in expressibility and stability in designed antibodies, we asked the question whether by chance or not by chance, we figured out a way to improve the stability, general ways of improving the stability of proteins by design. And this is quite important because although stability is a major problem in much of biochemistry, so even though this is the picture we have of how a protein folds, it emerges as a nascent chain from the ribosome and then folds into the native state, in practice, when we go to the lab and actually try to overexpress, especially a mammalian protein and a microbial host, this is often what we get, misfolded and aggregated species. And this has many, many adverse consequences for both basic and applied research. So the method that we developed, which we called PROS, provides a quite general solution to this problem of low stability and expressibility. I'll explain how this method works so that you can see how we think in practice about evolution-guided atomistic design. So we start with a multiple sequence alignment of the target protein and a set of its homologs. Here I'm just showing three positions of a given enzyme. And we rule out any mutations that are very rare in the multiple sequence alignment. We presume that evolution has tried these mutations and found that they either decrease activity or promote misfolding. So we eliminate these identities from further analysis. We then use Rosetta atomistic design calculations in order to introduce each mutation singly. And any mutation that destabilizes the protein is also thrown out, also eliminated. So now we're left with a much smaller tolerated sequence space. And it is this sequence space that we allow Rosetta design, combinatorial Rosetta design, to work with. And we design typically 3 to 7 different variants, each of them encoding dozens of mutations from the native state. This is the approach. Just a second. When we first came up with this idea, we asked Dani and Yoel what is the best protein to work on, and they recommended Humanistic Cholinesterase, which is Yoel's favorite protein. Yoel and Dani had worked for many years trying to express this protein in microbial hosts because it's a very important protein for biomedical research. But this enzyme is truly monstrous. It's more than 500 amino acids in size. It has multiple disulfide bonds, as you see here, in glycosylation sites. And its active site, shown here in yellow, is 20 angstroms below the surface of the enzyme, meaning that the enzyme is also very sensitive to mutations. So this is a well-known, very difficult problem, although people have tried for decades to make a variant of this enzyme that could be expressed functionally in microbes. Those efforts largely failed. So we designed 5 variants of this enzyme, again, each of them encoding dozens of mutations. The best variant encoded 51 mutations relative to the wild type, which at the time was simply unprecedented for stability design, and yet this design was actually the best in our set. If you look at well, it's difficult to look at all the 51 mutations, but if you look at some of these mutations in isolation, they make sense. The mutations are shown in green. They fill in cavities within the core of the enzyme. They rigidify the backbone or introduce new hydrogen bonds and electrostatic interactions. The really remarkable thing is that when you introduce simultaneously all of these mutations to this enzyme, you get really remarkable changes in biophysical properties. For instance, the bacterial expression levels, this is the bacterial expression level of Humanistic Cholinesterase in bacteria. You can see that it's basically nothing. The 5 designs express to more than 100-fold higher levels, with the one shown here with 51 mutations expressing to nearly 2,000-fold higher levels than the human in standard bacterial cells. The 5 variants are also 20 degrees more stable than the human variant, and Yoel's group solved the crystal structure of this enzyme with 51 mutations again, and the active site is virtually indistinguishable from that of the human enzyme, and accordingly, the activity of this enzyme is also nearly indistinguishable from the human enzyme. Despite 51 mutations, it's still a diffusion-limited acetylcholinesterase. So these results were a first for us. We never saw such robust and reliable design of such complex enzymes. And we next asked whether this method could also be applied to biomedically relevant problems. But you would appreciate that in most biomedically relevant problems, introducing 51 mutations is simply out of scope because of the potential for provoking an adverse immune reaction. The one area where adverse immune reactions or immune reactions are not a problem is in immunogen, vaccine immunogen design. So we turn to a problem for malaria. Malaria is still a very serious disease. You can see here the data. And despite 60 years of research, there's still no effective vaccine in the clinic. About a decade ago, a protein called RH5 was discovered and recognized to be potential for, very exciting actually, potential for a vaccine immunogen against this terrible disease because it's absolutely conserved in all field isolates of Plasmodium falciparum and absolutely essential for the process by which the parasite invades our red blood cells. This is a structure of RH5 as solved by Matt Higgins of Oxford University. So again, it's a very promising vaccine candidate, but like many other proteins from parasites and other pathogens, it's very difficult to work with. It degrades at about 40 degrees Celsius, and it can only be produced in very expensive insect cell cultures. And this is important because if this protein will ever reach clinical stage, as it is actually right now, it will have to be produced in hundreds of millions of doses and shipped to some of the poorest regions of the world where there is no electricity and therefore no refrigeration. So we need to identify a variant of this protein that could be expressed preferentially in microbial hosts to make it more economical and also much more stable. So we designed 3 variants of this protein, and we sent the sequences to our collaborators, Matt Higgins and Simon Draper at Oxford University, and we suggested that they might try to express these variants in bacterial cells. And they were initially quite reluctant to try that, because whenever they tried any construct of RH5 to express in bacterial cells, these are the expression levels they got, nothing. But the 3 designs, I'm very happy that they tried, the 3 designs expressed to very substantial amounts in standard bacterial cells, and this was incredibly exciting for them and for us. Design 1 was carried forward and shown to be 15 degrees more stable than the wild type. And also, they then injected wild type RH5 in our design into mice, isolated the polyclonal antibody response, and tested what is the immunological function of these antibodies. And you can see here that the neutralizing titers in both the wild type and the design are essentially identical. So just like in acetylcholinesterase, the biological function of this design is indistinguishable from that of the wild type, but it is a much better reagent to work with. And I'm very happy to report that a few months ago, this was licensed for commercial mass production. So this shows a very clear example for how an automated method can open up bottlenecks in protein production. So this is a completely automated method, and we want everybody to use it. So we developed into a web server, and you can see that over the course of the past 3.5 years or so, really thousands of people have tried it out, and there are publications that show that it works on enzymes and proteins that I've never heard about. So it's quite exciting, and to me this is a very appealing future for protein engineering. Instead of relying on iterative strategies, maybe in the future such automated methods could be used to reliably, accurately, and very quickly overcome bottlenecks in design of more stable and also more functional proteins. So with this, I come to the question of how do you improve protein activity? With PROS, we only design mutations outside of the active site of the protein because we only want to impact stability. We don't want to affect the protein activity. If you want to design more active proteins, you have to address the active site. And this is quite challenging because if you look at any active site, this can be also, I'm showing an enzyme here, but this can also be the binding site in protein-protein interactions. If you look at any active site, what you see typically are these long amino acid residues that form critical interactions with the substrate, and also, quite crucially, they form atomic interactions with one another. And you can imagine that if you mutate any one of these positions independently, you could cause a complete collapse of this crucial network of interactions among these residues, resulting in orders of magnitude reduction in catalytic activity. How does evolution address the question of improving protein activity? As I learned from Dani, we start from some low level of activity. We generate a genetic diversity, and then we select the variants that have improved activity. And these methods, as Dani pointed out, are very effective at optimizing protein activity, but they tend to get stuck, as you see here. They climb up mountains, but then you don't really know whether climbing up results in something suboptimal as this or the most optimal variant possible. And once you get stuck like this, you have to essentially rewind the process and restart it. We asked the question in this method called FuncLib whether computational design could jump over such fitness valleys between activity peaks. So we start the design process by freezing both the sequence and the conformation of the absolutely crucial amino acids within the active site pocket. These are the residues that are responsible for the catalytic step. And we want to diversify all of the other amino acids within the pocket. Now, crucially, in all of the enzyme design methods that I know, you absolutely need a substrate-bound or transition state-bound model of your enzyme. But actually, if you think about it, these models are often quite inaccurate. So here we asked whether we could get away with not modeling the substrate or transition state at all within the active site pocket, and we're just designing the ABO state of the active site pocket, essentially looking for constellations of amino acids that would be stable and pre-organized. And the underlying hypothesis here is that stability and pre-organization are some of the most essential features of high-efficiency enzymes. So we start the process similar to PROS, with a conservation analysis here only restricted to the active site pocket. We also use mutational analysis in Rosetta, again, to eliminate mutations that are highly destabilizing. And we're left with just a few mutations at each position within the active site pocket. But if you think about the combinations of mutations here, they can be quite large. I mean, if you multiply all of the combinations here with these, you have a tolerated sequence space that could encompass between hundreds to 1 million different variants within the active site pocket. And keep in mind that here we're not looking for the best variant. There's really no idea here, no notion of what is the best variant. We're actually looking for a diversity of different variants and whether they could catalyze the reactions that we look for. So we enumerate all the combinations of mutations seen at position 1 with all the combinations seen at positions 2, et cetera, et cetera. We model them in Rosetta, relax structures, and then rank them by energy, cluster the sequences, and then take, let's say, the top 20, 50 or so sequences for experimental testing. The first model system we worked on, again suggested by Dani, was a bacterial phosphotriesterase. This enzyme is a very broad spectrum, promiscuous esterase. It hydrolyzes this pesticide, but also lactones and various esters. The reason it's so interesting to us is that it also hydrolyzes organophosphorus nerve agents such as VX, serine, and so on. These are some of the most toxic and poisonous compounds we know, and one drop on the skin of an adult can kill within minutes. So there's a lot of interest in developing countermeasures for this, for these compounds, but the natural phosphotriesterase, the natural bacterial phosphotriesterase has activity rates that are far, far lower than is required for therapeutic use. So we used FuncLab in order to design about 65 different variants of the natural phosphotriesterase. And here I'm showing just the best of the set. You can see that the y-axis reports the fold improvement in log scale relative to the wild-type phosphotriesterase. And here we're testing against 6 different substrates, including these nerve agents, VX, Russian VX, serine, and so on. And you can appreciate that just with a few mutations, 3, 4, 5 mutations from the natural enzyme, we're getting 100 to even 4,000-fold improvement in catalytic activity relative to the wild type, including against these toxic nerve agent poisons. So even though these are just a few mutations, the selectivity profiles also vary dramatically from the wild type, which is shown here at the top. Notice especially the 2 enzymes at the bottom, which have very broad substrate selectivity against essentially all of the nerve agents we know. So this was a very exciting example for us because it showed that with this computational design method, we can achieve therapeutically relevant enzymes within a very, very short time. We essentially got these, Olga got them within just 2 months of starting the design process compared to potentially many months or years even for iterative in vitro evolution processes. So we can reach high activity peaks using computational design. The next question we ask is whether evolutionary processes can even reach some of these designed proteins. So why were we interested in this question? If you look at some of the designs, this is a crystal structure shown here in pink of one of the best esterases in our set. You can see that this 4-point mutation enzyme has mutations that are clustered, and they're medium to large from this histidine to tryptophan and from histidine to arginine. So we sort of presume that these mutations will show very complex interdependencies. So Olga turned to mutational analysis. In this plot, what I'm showing you is the relative specific esterase activity of the wild type shown here. It's very low. It shows very low esterase activity. each of the point mutations, each of the 4 point mutations, improves catalytic activity. This single point mutation does a very good job at improving catalytic efficiency. So if this were some sort of in vitro evolution or rational design process, you'd be very happy with these results. The next step would be to recombine these mutations. And this is where the disappointment comes. Each of these 6 double mutants is lower than the single point mutant, and 3 of them are even lower than the starting point Y-type enzyme. And yet, if you combine this double with this double, you get the quadruple design. And this shows very clearly how computational design may jump over fitness values in evolutionary trajectories. So at the time of developing this method, we were also developing a method for designing high specificity protein-protein interactions. I won't go into the details of this method, but we were successful in designing pairs of proteins that were highly homologous to one another and yet exhibited more than 100,000-fold specificity switches in binding their cognate partners relative to non-cognate homologous pairs. But some of these pairs were relatively low affinity. For instance, one of the design pairs had about 70 or 80 nanomolar affinity. So instead of doing regular affinity maturation, which can take months and months, and screening millions of different variants, we asked whether FuncLib, again, could apply and improve the affinity directly. So here you can see we tested just 19 variants. The best one improved the affinity from 70 nanomolar to sub-nanomolar level. Again, about more than 80-fold improvement in affinity through testing just 19 handpicked or computationally picked designs. So if we summarize this part of the talk, for quite a few decades, the leading method to optimize proteins relied on the iterations of maybe some rational design, but randomized screens. These iterations are quite laborious, and now it is possible to optimize at least some of the properties that are of interest to us in proteins through computational methods. Some of them are available through web servers. So in the next section, I'd like to go back to the question of backbone design. So I mentioned modular assembly and design with respect to antibodies. Here we asked the question whether modular assembly and design could also apply to enzymes. And we turned to Tim-Barrel-fold enzymes because of their modularity, this beta-alpha modular fold. So generally speaking, in many enzymes, you can classify them according to their molecular activity. And if you analyze their structures, enzymes belonging to a single family catalyze the same reaction. If you analyze their structure, what you see is that the core catalytic residues are highly conserved, essentially even in atomic details, whereas the periphery shows really vast changes, both in sequence and in conformation, including by insertions and deletions. And this is typically where substrate selectivity is encoded in enzymes. So we asked whether we could recapitulate this feature, this architectural feature of enzymes using computational protein design. So here we start again with the structures of natural homologs. We align them and we freeze all of the positions within the active site pocket. We then segment the enzyme into pieces. So we have many segments coming from a single family, let's say 20 different segments for each of these parts. And then we assemble these segments together to generate new backbones, just as I mentioned for antibodies. But unlike, let's say, gene recombination, which just takes specific genes and then recombines them, here we design the sequences of these segments for maximal compatibility so that we get, hopefully, stable and active enzymes. And finally, we also use Prof, of course, to make the designs more robust. The key point here, as in antibodies, that even if you have just 20, let's say just 20 unique structures for a given enzyme family, you can have access here to more than 100,000 unique backbone conformations. So this can provide a vast number of a vast amount of structural diversity, which may also translate to functional diversity. So the two Tim-Barrel fold families that we targeted here were one, phosphotransferase-like lactonases, which I introduced earlier, and a second, which was completely unrelated in function or in sequence. These are xylanases. I think Gideon Davis will talk much more about these than I will. I'll just say that these enzymes are critical for biomass degradation. So again, two unrelated families. If you look at the designs coming out of the design strategy, what you see is that they contain many, many mutations relative to the closest natural homolog, typically 50 to 150 150 mutations. And there are many gaps between, again, the designs and any close homologue. One of the reasons why we chose xylanases and PLLs for the study is that for both of these, there are well-established medium throughput assays to improve these designs. And we thought that, similar to all previous enzyme design studies, that the designs that will come out of the method will be sort of mediocre and that we would need to carry them through in vitro evolution. But really, to our great surprise and excitement, in this case, the designs coming out were highly functional compared to natural controls. Some of them were actually a little bit more functional, more active, than these natural controls. And this was the first time that we saw a completely automated method, design method, produce such highly active enzymes. You can also appreciate that some of the enzymes have different selectivity profiles. This enzyme is a pretty good paraoxinase, whereas these enzymes show absolutely no paraoxinase activity. They were also quite stable essentially as enzymes coming from extremophiles. We also saw the crystal structure of one of the most high-efficiency xylanases, and as you can see here, all of the contacts were essentially, as we designed, it's quite boring comparison between the design structure and the actual crystal structure, especially within the active site pocket. It's essentially identical to the design conception. The loops are in place. Everything is in place. So this was very encouraging for us. It provided a very strong validation for this modular assembly and design strategy. So for the last part of my talk, we asked the question, given that this previous method was completely automated and generated functional enzymes, what if we now design not a few dozen different enzymes, but really vast repertoires comprising millions of different enzymes, as Madan alluded to before? And why is the reason? Why would we want to do something as crazy as that? The reason is that given so many designed enzymes, if we can effectively screen them, we may be able to learn what features discriminate active from inactive designs. And that may really, in a systematic way, improve our understanding of how to effectively design new enzymes. So here, we really took a leaf out of a standard immunology textbook. We're really inspired by antibodies, as you could see throughout the talk. In antibodies, the high versatility of antibodies is encoded in short DNA fragments, which are recombined. Here, we're essentially asking the question, can we design fragments of enzymes that could be recombined with one another to generate a huge set, millions of enzymes again, that would be stable and functional. So how do we do this in practice? We start with a computational step. We're very similar to the previous section. We align xylanases again, members of the same family. We segment them, and we use PROS and FuncLib both to stabilize each segment on its own and also to diversify the active site pocket in order to generate more functional diversity. Now we move to the test tube, and we synthesize a few hundred different fragments of DNA for each of these fragments that we designed here. We then assemble them in the test tube to generate 1 million different xylanases, and we transform yeast for yeast cell surface display. How do we functionally screen so many enzymes? Here we use, and again, I think that Gideon will talk about this at much more length, we use an activity-based probe. This is a Xylan analog, and this activity-based probe can label active xylanases. It binds to the active site of the xylanase and reacts with it, but isn't released. And now it's fluorescently labeled, so we can use standard FACS sorting in order to isolate the active from the inactive designs. Now we use deep sequencing in order to determine which were the active designs. And finally, we use, as again Madan alluded to, machine learning methods in order to understand what are the features that discriminate active from inactive designs. So from this crazy pipeline, we got 4,500 unique sequences, unique design sequences that were active according to this assay. And you can appreciate that this is a very small subset of the structures. They come from 700 unique backbones. So a huge diversity of backbones. You can see that the active site is really highly mutated, including by insertions and deletions. In fact, if you cluster the sequences or the structures, according to structure clustering, these are the structures from the PDB. They form one clade within the structures that we designed. So we're essentially expanding the structure space for this family tremendously by using this strategy. And this is important because, again, structure diversity may encode functional diversity, as I'll show in just a few slides. So what can we learn? Again, the main purpose of this study is to learn new features that are important for discriminating active from inactive designs. So we can use a variety of different structure and energy features to discriminate these designs. Some of them are very interesting for us. For instance, hydrogen bond energy is much improved in the active designs versus the inactive designs, et cetera, et cetera, et cetera. There are about 9 or 10 such features that discriminate them, but you can appreciate that the distributions are highly overlapping. But now, by the magic of logistic regression, if you combine these through a logistic regressor, what you find is a really complete, almost complete discrimination of active from inactive designs. So this seems like a very powerful separator of active from inactive designs. And now the question is, can we actually use it to improve the repertoire? So Rosalie, who made this project possible, went back to the design procedure, generated another 300,000 designs now selected according to this logistic regressor, and tested their activity again with these activity-based probes. You can see here that now she's getting a very active and very stable repertoire of xylanases, but not only xylanases. Again, we got another activity-based probes from Herman Overcleft, this one for an analog of cellulose. And you can see that even though the original xylanases are essentially inactive towards this cellulose analog, we're getting a fair amount, a large subpopulation that is active against this cellulose analog. So in fact, we can expand the substrate selectivity using this strategy. So this is quite exciting for us, and this slide essentially summarizes everything I showed you. And this is really the paradigm that I find very attractive in protein design. We start with some hypotheses about how to design, what are the essential features that need to be encoded in proteins in order to get effective designs. This hypothesis can be incredibly naive, as I showed you for antibodies in the beginning of my talk. Nevertheless, we design a bunch of designs, we go to the lab, build them, test them, and then we can learn from our mistakes. And crucially, we can go back to the design procedure to verify that what we've learned is general. And as I showed you here, we're making progress. We're still not at the point where we can design antibodies, for instance, from scratch against very challenging targets. But as you could see from this procedure, we came up with methods that are very effective at optimizing both antibodies, other binding proteins, and enzymes. With that, I'll close. Thank the lab. This is a very old picture. Half of the people have left. Another half have joined. I'm sorry about that. But I thank you for your attention.
So thank you very much, Sarel, for this talk. I think it's really quite amazing what comes out. And it I think, shows that biology has become a data science, information science, plus going back to test. Yes. So the talk is open for questions. Carol.
That's great. I'm one of your academic users, as you know, so I can attest that it works really well. We had several Prof solutions, though, and there seemed to be a very high number of changes that had to be made. So I was wondering if there's scope to compare Prof solutions and then reduce the number of mutations that have to be made and still keep the high expression level.
It's very difficult to answer this question. Deconvoluting dozens of mutations and finding out which are the most essential ones is quite difficult. We've never succeeded in doing this. What I can say is that if you really require just a small subset of mutations, FuncLib, I didn't show data on this, but it does seem to stabilize proteins as well. If you apply it, for instance, to the core of the protein or to obligatory interfaces, for instance, in antibodies, you get very high expression yields and stability. So it's another approach to stabilize proteins which has less, fewer mutations. Yes.
Okay, thank you.
Way back,
That was wonderful, thank you. In these proteins engineered to express in bacterial systems, what has happened to what were the glycosylation sites? Because if you're a glycobiologist, you'll have been taught that the glycosylation is initially part of the folding process itself.
Yes.
And if you have glycosylation mutants, the proteins don't fold, not that they fold and are insoluble or whatever, they just don't get. So what solution have you found to what was the folding use of a glycan?
So this is a really excellent question. Both in acetylcholinesterase and in RH5, there are multiple glycosylation sites. I can also say that there's another enzyme we worked on, a human enzyme related to disease, where glycosylation was exactly as you say. It was thought to be absolutely essential for folding and stability. And yet the enzymes in all of these cases, or the binding proteins, were expressible in bacteria. So I can't say for sure what's going on. We typically conserve the, let's say, asparaginins involved in glycosylation because they're absolutely conserved in the multiple sequence alignment. So we don't allow mutations in absolutely conserved positions. It's just that we're stabilizing everything in other parts of the scaffold of the enzyme. So I would say that maybe we don't actually know the answer, but I would argue that within human cells, glycosylation provides a way to optimize the expression levels and stability of enzymes which are sort of not so great. But there are other ways. They may require many mutations.
Okay, thanks.
I think it's okay. So we can, again, thanks, Sarel. Go to the